Hello, this video is intended for clients of the UCLA Lipidomics lab. In this video, we will walk you through the sample submission form. This Excel workbook allows customers to provide detailed information necessary for the analysis of their lipidomics data. Filling out this form properly ensures that the final data you receive is accurate and easy to interpret. It can be downloaded from the UCLA Lipidomics website. It should be filled out and emailed to us upon sample submission. The Excel workbook contains 2 sheets. The map sheet is for you to fill out. The other sheet provides an example. Let's take a look at the example sheet first. The top left table is an autofill summary of your experiments. Please do not edit this area. To the right is a form to fill out with contact information for the person who will handle your invoice. Usually this goes to the PI, but it may be sent to a lab manager, admin person, or a researcher in your lab. In the form below, you will fill in your sample information. Please do not edit the first column or the column titles. You should avoid deleting or inserting rows or columns. Also, try to avoid using any special symbols as you fill out this table. Now let's go over the information you will provide. The first 2 columns next to sample number are experiment number and experiment name. These are used to parse your data into separate Excel files and plots. Sometimes you might want to parse samples into separate experiments even if they are considered one experiment in your project. For example, here we show a plot of an experiment that includes both cells and adipose tissue. The scale of total TGs measured is so different between these 2 types of samples that it is difficult to interpret the plot. Additionally, the normalization factors are different between cells and tissue. Please note that the experiment number should be ordered, starting from 1 and going forward. The experiment name should be the same for each sample within a given experiment number. We recommend that you use simple names to make your experiment easy to identify. The next 2 columns are group number and group name. Within each experiment, the group number should start from 1. Groups may be biological or technical replicates that you wish to compare against other groups within the experiment. As an example, a group may consist of biological replicates of cultured cells that have received a drug treatment, or the livers from several mice with a given knockout. Unlike the experiment number, the group number doesn't need to be ordered and can be mixed up in any order. The group name should be the same for each sample with a given group number. These are used to parse out data to compute group average, variance, and to make plots. The groups in the class total plots are ordered by group number, so you can assign group numbers based on the order you want to see them in the plot. The next column is sample ID. Each sample should have a unique sample ID. The next 2 columns are sample normalization factor and normalization type. The common normalization factors are milligrams for tissue, cell counts for cells, and volume for plasma. If you wish to submit samples with 2 different normalizations or 2 different sample groupings, you should create a second sample submission sheet with identical sample IDs and the alternative sample information. And make sure to mention this second data analysis scheme in the email in which you submit the 2 Excel sheets. Let's move to the map sheet. As you fill out this form, the cells may turn red to alert you to possible problems. You can ignore these alerts until you finish filling out the sheet. Once you have finished, if there are still alerts, please go back and check for mistakes. For example, in this form, the group name for sample 7 has an extra space in the end. Once you remove the space, the alert should disappear. Note that in this example, the group numbers are not ordered. As previously mentioned, this is allowed for group numbers within experiments, but is not the case for experiment numbers. The experiment number has to start from 1 and remain ordered. Once you fill out the form, name the Excel file in the following format: date, lab or company name, batch name. Then email this form to us when you submit your samples. We highly recommend that you label your samples with the sample number in the first column. When submitting cells, the sample rack has numbers seen through the bottom of the collection tubes that should correspond with the sample numbers in the first column. For tissue or plasma and serum samples, the respective tubes should be numbered with the sample number in this first column. If you have any concerns or problems after reviewing these instructions, please reach out to us for assistance.